Where's it out of? Rockport. Okay. Here in Texas. All right, let's see here. Buffering. Don't have anything else open. Oh, there it is. Good. Excellent condition. All right. New day. New 8 a.m. class. I sent out the announcement on Monday because I forgot to tell you. When we finish that presentation on dispersion and population size, I have a quiz that's posted. So get that done. I made that announcement on Monday. And of course, as soon as I got back in my office, I realized I forgot about it. But it's posted. Get it done. We have lab next week. Bring calculators, bring your laptop, tablets. We're doing life tables. We're filling out life tables. This is something you could do by hand. It's also something you could do on the computer. We'll work through a life table. Try to fill out a practice one, and then you could do it on your own. And then there is some homework that has you doing more of those tables. Again, with those life tables, because they take some time, not really, I can't really ask you to fill out a life table on an exam, but I could give you a life table, all right, and tell you to calculate some things from it, or I could ask you maybe what are the variables? What are the variable names? We have on Thursday, just so you know, I won't have office hours in the morning, so I'll just put a sign on my door. Probably can respond to emails, but it'll be sketchy. I'll get back here. I should get back in time for lab. All right. So, survivorship curves. So with life tables, one of the things that we can do. Yeah, we got the full one. One of the things that we can do, one of the utilities of these life tables, is to generate a survivorship curve. This survivorship curves tell us the age-specific survivorship, hence its name, survivorship curve. All right. From this survivorship and mortality go hand in hand. So if we know what the survival probabilities are, we know what our mortality rates are going to be. All right. Now with our survivorships, these values are the probability of getting to a specific age category. All right. Not just a survival for this year to make it to next year, or survive next year to make it to the year after that. It is survival from birth until that age category. All right? And that tells us a lot of information about how our organisms may reproduce, their strategies and how many offspring to have, and whether or not they should have parental care. Things like life history type of stuff, which we'll get to. We'll talk about. So with our survivorship curves, we have 3 different types. We have our type 1 curve, characteristic of humans. All right. You have initial, higher mortality, elevated mortality is probably a better word. Elevated mortality rates in the very young. All right. These are the newborns. These are the very young kids. All right. And then once they escape that early stage, then what happens is they have high survivorship throughout most of their life. So, if we looked at, let's say, probability of surviving to reproductive age, we have a pretty good chance at doing that. Right? If we survive that early stage, then we have a pretty good chance of surviving to reproductive maturity. And then once we get through reproductive maturity and reach our lifespan, or end of our life, then mortality rates pick up, our survivorship drop off. All right? Type 2 patterns, birds. All right? We have a constant mortality rate. So we did our mortality curves. We ended up with those. So if we looked at our QX, which is our mortality, this is age. Our type 1 curves look something like this. Our type 2 is just a flat line. And if we have a flat line, constant mortality rates, then we get this steadily decreasing survivorship curve. All right, so the chance of dying is the same this year as it is next year, and so forth. We don't have the elevated mortality rates. Now, I did say for birds, you do tend to see 2 different curves, one for the nestlings and one for the adults. All right, but both of them tend to be straight. Both of them tend to be straight like this. And then our third type of survivorship curve is this one. All right, trees, invertebrates. All right, we see just a pretty rapid drop in survivorship and then a flat line. All right, in this case, we have high mortality rates early in life, and then once we make it to adulthood, then mortality rates are much less. So you can kind of think about fish or aquatic inverts. All right? Fish have a huge number of offspring. Most of them die. Why do they die? What gets them? Yeah, everything. Predators, all right? They're susceptible to changing conditions. Water rises up. They get stranded on the shore. A huge number of them die. They have a high rate of mortality early on in their life, but if they make it to reproductive maturity, if they get to adulthood, then your mortality rates are going to be pretty low. All right? So these are 3 types of survivorship curves. These are the mortality curves that go with it. And just remember, our survivorship, L sub x, our graph is on the log scale. All right, so why do we look at survivorship curves? Well, it tells us a lot about this, about the mortality rates. But we can also compare survivorship curves because it could reveal important biological processes. The things that we're comparing are the slopes of the line and whether or not our lines are parallel. All right? So if our lines are parallel, then it means that we have similar mortality rates. Yeah, they're at different elevations, but what changed that is how we got there. So earlier in life produced those differences. If we're parallel, then we have similar mortality rates. If our slopes are different, we have different mortality rates. All right, so here's an example. These are 2 different deer species. All right, Otocoilus, we've got populations that live in the chaparral, populations that live in the shrubland. I want to say I think these are California populations. It's an example that I believe is in your book. And what they did was generate survivorship curves. Now this is on the log scale. Even though it says L sub x, this is the log scale. All right? So what you see is that survivorship actually appears higher on the chaparral region than on the shrubland region. All right? And what you see then is that our lifespan's the same for these deer. They live about 10 years. That's about as long as they'll live. We have higher mortality rates in the shrubland than in the chaparral region. So higher survivorship means lower mortality rates. And we have different slopes. So one, they're not parallel, so there's a difference in these habitats. And that difference is something that's affecting our mortality rates. Perhaps the shrubland is harsher. Perhaps it's less food, less resources. Maybe another predator. Maybe there's a big top predator in that area compared to the chaparral. That's the things that we would look up. These are the things that we would then pursue to see what's causing this difference. This one, Capreolus, another type of deer species. It is, who knows? It is, hold on, I'm thinking, gonna look it up. It's a European deer species. Roe deer. Slide that up. Here it is. Roe deer. I knew that. Tip of my tongue. What's that? I don't know. Don't know. All right, so second, survivorship. Now this one is male versus female. This first one with Otocoilus was just total population. Capreolus, male versus female, and you see we have a difference. All right, we get divergence later in life. So right on Otocoilus, you get divergence almost immediately, suggesting that there is a difference in mortality rates that's between these two habitats. Here, we don't really get our divergence until a little bit later in life. So our mortality rates early on seem to be about the same, and then we get the divergence between male and female. Now these parts are parallel, which means in that early age category, we're experiencing similar mortality rates. But then you get sharper slope, steeper slope for males than for females. And that sharper slope suggests that we have higher mortality rates for males. Now, why would that be the case? Why would we see higher mortality rates for males? What's that? Say what? Hunter preference. Okay, that's a possibility. Yeah, definitely hunter preference. That can have an effect. What else? What happens about year 3? So even though it says year 2, that's 2 years of life. They're in their 3rd year at this point. What happens about their 3rd year? I should say, what doesn't happen in that 1st year of life? What's that? Mating, reproduction. It's a big part. Big part. All right. So here, you don't see that effective mating because the males are going to compete. Males are going to try to compete for mates. They're going to try to compete for some territory. All right. You don't see that difference, that effect, until we get to reproductive age, and then it's pretty significant. All right. So we can identify habitat differences, difference in sexes, especially mortality rates. These differences in mortality rates just by looking at these survivorship curves. And we'll do some of them. Yep, competition with other males, antlers, yep, fighting each other. It's all of those things that are affecting these differences. All right, so survivorships, that's number 1 for our life tables. Second utility of our life tables is life expectancy. All right, so life expectancy is the mean expectation of further life for an individual of age X. The mean expectation of further life for an individual of age X. It's how much longer can we live? All right. We don't want to confuse this with lifespan. Lifespan is just the maximum age to which we can live. All right, and that's going to be basically species specific. All right, human maximum age, you can say, what is it, about 100, maybe 95? And then you've got variation, individual variation that's going to change that number. All right, that's not our life expectancy. Our life expectancy is how much longer we expect to live. All right, so the life expectancy of a newborn is going to be different from the life expectancy of a 20-year-old. All right, because a newborn has to survive all of those early years. So it's going to take a hit on its life expectancy. But if we can get through those years and make it to until we're 20 or 21, then our life expectancy could predict that we live much longer. All right? Same thing would happen, let's say, with the type 3 survivorship curve. The life expectancy of a fish when it's born isn't very long. But if that fish survives that first year, or maybe the second year, depending on the species, then our life expectancy could be several more years after that. All right, so the life expectancy is the mean expectation of further life of an individual of age X. The life expectancy of a population is going to be calculated for those newborns, because we want to include all sources of mortality at all ages. But the life expectancy, you can do it for every single age. So what affects our mortality rates? Survivorship. So QX and LX. LX is our survivorship, QX is our mortality. If we increase our mortality rates, if we increase these Qs, we decrease our life expectancy. And that Q can happen at any of our ages. All right. The flip side, so if we increase our Q, then we decrease our L sub X. All right. If we decrease our L sub X, we also decrease our E sub X, our life expectancy. If we decrease our Q, then we're going to increase our survivorship. And if we do that, then we increase our life expectancy. So survivorship and mortality rate, they do affect and determine our life expectancy. But there's one more thing that affects our life expectancy, and that's lifespan. If we can increase our lifespan, then we also increase our life expectancy. We add more years that we could potentially live because our lifespan got longer. Is lifespan genetically determined? You say yes? Okay. Who says yes? Yeah, it is. It can be. All right. I do think when we talk about life history traits, I mentioned lifespan, but yeah, scientists have demonstrated in the lab increasing lifespan, selecting for individuals that live longer. So yeah, it could be genetic. So if evolution is increasing our lifespan, then we can increase our life expectancy. Decrease the lifespan, we decrease our life expectancy. Any questions on life expectancy? We'll calculate this from a life table. You'll see how we do it. All right. And the third thing, so I think survivorship is very important. I think life expectancy is maybe it's important, maybe it's not. I think population growth rates, it really is one of the main drivers for doing a life table. All right. Because we're studying a population, we want to know, is this population growing or is it declining? So once we have our life table, once we know our age-specific mortality rates, and we know our reproduction, our age-specific reproductive output, then we can calculate a population growth rate. All right? So we need to know our chances of survival to reproductive age. We need to know our survivorship. And then we also need to know this reproductive output. And as you're going to see in the lab, B sub X is our reproductive output. If you did a search, it drops again. All right, so it can change throughout the age categories. For dioecious species, so those species that have males and females, this birth schedule represents the number of female offsprings born to a female. And this is important to note because when we do these population growth rates for a population that's a dioecious species, we're really only including females in those tables. All right, we're only including these female individuals in that life table because females are the only ones that are going to be reproducing. So that's our target. So this birth schedule is our expected reproductive output at age X, and we use the B sub X variable for that. And I'm going to pause here. So I'm seeing we have some connection issues happening on our live stream. It doesn't appear to be on my end. I think it's the internet here on campus or going out to YouTube that's having some issues, so apologize for that. All right, so anyways, population growth rates. We have to account for these two things, and we account for them by multiplying our BX and our LX columns together. All right, that's going to give us something called realized reproduction. This realized reproduction, so I should step back and say birth schedule gives us the expected reproductive output, but that's just the expected. That's for those individuals that have already survived. If we are interested in knowing what our population growth rate is, we have to account for that possibility that individuals do not survive to reproduction. All right? So what we do is multiply this LX and BX columns to get a variable that I'll call realized reproduction. All right? I put realized in quotations because it's not really necessarily a thing, but it kind of gets across this idea that our individuals have to not only survive, but they also have to reproduce. All right? And that's important. It's important when we predict population growth and decline. All right? So once we have that LX-BX column, what we do is add them all up, because this is our realized reproduction for each individual, for each age category. And then when we add them up, that gives us basically our realized reproduction for the entire population. Now this realized reproduction for that entire population has a name, and it's called net reproductive rate. This is R naught, R sub 0, R naught. You've probably heard about R naught in the news. It was all over in the spring as COVID was spreading. All these journalists kept using this big fancy term R naught, and they probably had no idea what it meant. All right? But it's talking about transmission in the case of the viruses, or for our freestanding populations, it talks about reproduction. It talks about how fast population is growing, or how quickly it's declining, or perhaps how it's not changing at all. So our net reproductive rate is the average number of offspring produced by an individual in their lifespan. It takes into account survival, it takes into account reproductive output. All right? We calculate it from a life table using this R naught, and the sum, or we calculate it using the sum of that LX-BX column. And we'll do this. Our values are going to range from basically all around 1. Why is that off? Let me fix that. An issue of copy and paste. There we go. Save that. There we go. That's much better. Hold on. I have 2 population growths on here. Population decline. All right, that's better. So our R naught's going to range around 1. All right? So if R naught is equal to 1, then we have population stasis. It's neither growing or declining. Our population's staying the same. And the way you can think about it, since this net reproductive rate is the average number of offspring produced per individual, if our individual is producing an average of 1, then they are basically replacing themselves in the population. And if every individual replaces themselves in the population, then our population is neither growing or declining. If our population, if our net reproductive rate is less than 1, then we have incomplete replacement. Instead of replacing ourselves fully, we're only, let's say, half reproducing ourselves. And if each individual is only half replacing themselves, then over time our population is going to decline. If R naught is greater than 1, then we have more than complete replacement. Now we not only replace ourselves, but we can potentially replace somebody else in our population. And over time then our population grows. Okay? So our R naught's going to be around 1. If it's less than 1, we have population decline. If it's greater than 1, we have population growth. And if it's equal to 1, then we have population stasis. Now, you might be thinking, some of you might be thinking, how do we factor in males if we have a dioecious species? And I'll say, we don't. We don't. We assume that if the females' net reproductive rate is, let's say, greater than 1, then the males will also be greater than 1. And as we talk about for our sex ratios, females are the ones that are important. If we have more females, we're more likely to grow than if we have more males in our population. So here's a question. How can we increase our net reproductive rate? Let's say we're interested in a population, conserving a population. We've just saved it from the brink of extinction. Now we want to increase our population. What are some things that we can do to increase this net reproductive rate? What are some things that we can do? How would removal of predators affect our net reproductive rate? Decrease our mortality rate and thus increase our what other is there? Mortality and survivorship. Survivorship. So it goes back to R naught. Yeah, so one idea was to remove predators. If we remove predators, we decrease our mortality rate and thereby increase our survivorship. If you increase your survivorship, you can increase your R naught. What's another way in which we can increase this R naught? What's the other variable that affects R naught? Births? So if we can increase our births, we can also increase our R naught. All right, so survivorship and births, that's what affects our R naught. If we increase survivorship, we can increase population growth. If we increase our reproductive output, we increase population growth. All right. Now you can say, well, how can we possibly increase reproductive output? Well, it comes down to the species. If our species is one where if they have excess energy, they invest that into more offspring, then that's what we do. We might supplement food source. Supplement the food source so that they have that excess energy to produce more offspring. Not every species is going to respond that way, which is why we would have to focus on survivorship. All right. Yep, increased habitat quality came up as an answer, and that's exactly it. If we increase habitat quality, we increase survivorship. All right, so demographic characteristics. We talked about age structure, and the age structure is all tied to these life tables. Second demographic characteristic is sex ratio. Sex ratio is the proportion of a population that is male or female. You can present it as a proportion male, you can present it as a proportion female, you can do kind of as a ratio, so females to males or males to females, or you can say, you know, percent. We have percent females. All right? So how we phrase our sex ratio is just up to us. But the sex ratio tells us the ratio of males to females in the population. Now for polygynous species, these are species where the males mate with more than one female. Our sex ratio can indicate population growth or decline. All right. What do we mean by that? Well, it's this. The females are the ones that are giving birth. The females are going to be the ones that replace themselves and the males in our population. If we have more females than males, then we can see faster population growth because you only need one male to reproduce with all of the females potentially in our population. All right, let's say we have 10 females to every one male. Well, that one male could reproduce with all 10 females, and all 10 females could then mate and reproduce and produce offspring. If we have more males than females, then we have a slightly different situations. All right, all females might still reproduce, but now we have some males that do not. And this will suggest that we'll have a population decline because those males are unable to reproduce, unable to get replacement in that next generation. All right. So with our sex ratio, it all comes down to that reproduction. More females we have in our population relative to males, the faster our population can grow. Now our sex ratios will change as our organism ages. All right? So it leads to 4 different types of sex ratios that we can report. We can report a primary sex ratio, or a secondary, or a tertiary, or a quaternary. All right? So our primary sex ratio is that sex ratio right at fertilization. So if we're dealing with species where they all mate at the same time, they all give birth at about the same time, that primary sex ratio is the sex ratio that we would see once all of our embryos are fertilized. And basically they get fertilized, and then we kind of freeze frame it until all the mating in the population is done. Secondary sex ratio is that sex ratio that we see at birth. Why would they be different? Why would they be different? Did you say it? Say something? Why would they be different? What's that? Yeah, not all of them can survive. All right? Especially if we have some survival differences. And you can say in humans, you know, in order to be male, there's a sequence of development steps. There's pathways that we have to succeed in order to develop female. If any of those go wrong, then it may default to appearing female. All right? So, and you know, maybe males have a higher, male embryos have a higher rate of death before birth. All right? Tertiary sex ratios is that sex ratio at sexual maturity. So secondary ratios are at birth, and then from that time until we hit reproductive maturity, we have the possibility that our sex ratios can change. So we can report on the tertiary sex ratio. And that's right at sexual maturity. And then the quaternary sex sex ratio is a ratio of in our adults. So these are basically all of the reproducing and non-reproducing adults that are in our population. So we had already asked, why do they change? Why does it change from primary to secondary sex ratios? And we said it was mortality rates. Those changes in mortality rates, especially if there's a difference in mortality rates between the sexes, is what's going to be driving these changes. So going from primary to secondary to tertiary to quaternary, we're going to see those changes. And it's one reason why our tertiary sex ratios and our quaternary sex ratios tend to be skewed more towards females, especially in species where we have competition for mates. The males are going to compete. They're going to be exposed more to predators. They're going to have higher mortality rates during that period leading up to reproduction, which means that our sex ratios can start to be skewed. Primary sex ratios tend to be about 50-50. 50 male to 50 female. And then we start getting the skew. Now, so why do these major ratios change? We answered that, changes in mortality rate. Which of these ratios is most useful for predicting population growth? What do you think? Primary, secondary, tertiary, or quaternary? One, 2, 3, or 4. Which one? Quaternary? So I've got 1 vote for quaternary. We've got 1 vote for tertiary. 3 votes? 3 votes? 4? Tertiary is going to be most important. All right, because tertiary is right at sexual maturity. All right, that's where the bulk of our population growth is going to happen. All right, this quaternary sex ratio, while it may give us some idea of population growth, it includes individuals that have passed their reproductive prime and in many cases are no longer reproducing. So the most important ratio for predicting that population growth, it's that tertiary, right when we're at sexual maturity. All right, so what can affect our sex ratios? We've already mentioned differential survivorship, but I'm going to break this down into the genetic factors and then ecological factors, because these are kind of the most important things. So first up is just genetic factors. And what we're talking about here is that genetics determined if we were going to be male or female, XX, XY, all right? But that's only part of the story, all right? That's only part of the story, because you do have the regulatory pathways that have to operate. So while you can be genetically male, physically you could appear female. I guess you can also go vice versa as well. These genetic factors are going to affect the primary sex ratio, that ratio at fertilization. And then after that, then a lot more stuff can happen. If you're interested in more, check out the microteen rodents. These rodents that have a mutated X, all right, that alters these sex ratios even more, if you're interested in looking up more of that. So genetic factors, we're just going to say XXXY, that's our primary sex ratios. It's these ecological factors that then start to get us into the skew. So we mentioned differential survivorship. And generally speaking, our males are going to have higher mortality rates than the females, and it's for the reasons that we've discussed. They're going to be out competing for mates. They're going to be out competing for territories. That competition, fighting with other males, increases the risk of injury, but it also exposes them to more predators. All right? And because of this differential survivorship, we typically see this tertiary and quaternary sex ratio skewed towards females. What happened here? Okay. This did not pop up. We'll leave it here and then I'll pull it up. One of the figures, I have another graph here that isn't showing on this screen, but it's on my slide. So that's odd. Anyways, differential survivorship. I think probably one of the more interesting aspects is the environmental variation. So we also have the nurture component of phenotypic variation that can affect our sex ratios. And we've got two examples. All right. First example is this jack in the pulpit, Erythema triphyllum, this guy. This flower senses its environment, senses its quality of the environment, and has the ability to shift the likelihood of producing female flowers versus male flowers. All right? So what we see is in good conditions, these plants tend to produce more female flowers. I don't know why that is not showing up. So this is the figure that should be up, and we're only getting part of it. These figures are on the handout, so they're there. So the study went out and they looked at this trillium, another species, to see the percentage of browsing that occurs on that plant species. So what ends up happening is that the more that it gets browsed, all right, the lower the soil quality, the lower the nutrients in that soil. So they see this relationship that the fewer this trillium here that was present in that forest environment, the worse off the soil was, which meant then that our sex ratios began to be skewed towards our male flowers. The same exact thing happens if we compared these plants grown in the sun versus in the shade. Those flowers that have received much more sunlight are more likely to produce female flowers than those plants that have been grown in the shade. Now, why is that? Why do we see this difference? Well, it comes down to the energy requirements for these female flowers, or for the male flowers too. So the cost benefit. Female flowers cost over 3.5 times more than a male flower. So if you're in a poor quality environment, if you are not in the sun, you are basically energy deprived. You don't have the energy necessary to produce that female flower, yet you still want to reproduce. So what do? You produce the male flower instead. All right? If there's not enough nutrients, why would you invest all of that energy and time into a female flower on the chance that that female flower isn't going to reproduce or isn't going to successfully produce any seeds? It doesn't make sense. So only produce these female flowers when the conditions are favorable. Some turtles and alligators do something very similar. I know herpetology is being offered in the spring, or it's on the books to be offered in the spring. You'll probably talk about this in that class. You've probably talked about it in another class too. But what am I saying with this? It comes down to incubation temperature of those eggs. All right? If you have warm incubation temperatures, you're more likely to produce female offspring. If those eggs were in cool temperatures, you're more likely to produce male offspring. So what you end up getting is different sex ratio in those nests depending on the temperature of those nests. Where do we see those changes in the temperature? Well, consider this. What's that? Maybe a little bit, yeah. So this actually came up because people were interested in looking at sex ratios in turtles. And the easiest way to go about doing that, they were trying to get sex ratios as soon as they were born, right? So what they did was went out to a lake, get this zoomed in, went out to a lake and found nests on the shore. All right, here's my cattails and stuff, all right? We got different areas of the lake, and you have a nest there, and let's say we have a nest here. All right? The wind blows across the lake, right? What drives the wind currents? That was on, yep, okay, the spinning of the earth, right? So we do have natural wind patterns, directional wind patterns. So if we have wind that blows, what's going to happen is that we have this vegetation and trees and stuff protecting this part of the lake from the wind, and then as we get out to the open water, the wind blows closest to the water, hits the reeds and stuff, blows into the reeds before the wind kind of disperses. Say that's my drag. Very bad drag. Don't show physics that. All right? But what we get, warmer temperatures over here because it's sheltered from the wind, cooler temperatures in that nest. So what we end up seeing is that nest that's downwind generally produces more male offspring than females. This nest up here generally produces more female offspring than male offspring. This is a situation where genetics, evolution, has favored the development of the sensing system, where our developing embryos are sensing the environment and saying, hey, if I'm in a good quality environment, and female offspring and reproduction is more costly, this is when I want to invest in them. I want to invest in them when the environment is good. If the environment isn't looking so good, hey, let's invest in that lower cost male offspring. Because, as we're going to talk about, reproductive failure for a female is pretty bad. It's pretty bad for our population. But the male, hey, if it has reproductive failure, if it tries to mate with an individual and fails, that male just goes and finds another individual and tries to mate with them. Females can't do that. And especially with our organisms, where we have a very clear window where that reproduction and mating events are going to happen, if they fail that one time, they have to wait another year in order to reproduce. So our environment can influence our sex ratios. All right, what we're going to do is stop here. We're going to kind of wrap up with a summary, finish out with plants. Plants are really difficult when it comes to sex ratios. But we'll finish this out on Friday, and then we'll move to population dynamics. All right? So don't forget about the quiz.